Okay, well, top of the day to you, gentle people. I hope everyone's staying safe from the virus and that the economics of the situation aren't too dire. This is going to be really different, and I want to talk a little bit about risk assessment and management and regulatory efforts, because in many respects, our group of scientists, and I can tell you the regulators, are behind in development of methods to address the kinds of data that we just saw presented. So I'm going to give you a few slides out of what we do. We do a week-long boot camp where we're talking about risk assessment. I'll give you a few slides here. If we can go to the next slide, we'll talk a little bit about uncertainty analysis, and then we're going to talk about more efficient testing strategies. So the next slide there, Barry, please. So you can look at uncertainty analysis in many different ways, and there are many different approaches to do this. This is just one approach of many that comes out of the World Health Organization, and of course it's a framework, and the framework is very general. The first idea that comes from this is that there are uncertainties everywhere. There's uncertainties in the science, and we've just heard some of those spoken with our prior three gentle people. There's certainly uncertainties in the policy-based management decisions. These are imperfect data sets. If you think science is imperfect, then there's a lot to be said about that and to improve it. Policies are very different too, and they have different imperfections. And then, of course, risk communication. I find myself trying to do more risk communication all the time, and it's legion with the numbers of misinterpretations and misappropriations and just confusion when you try to communicate to people. And you add on to the fact that much of our public, at least in the US, is not scientifically literate. So if you hit them with all sorts of LLNAs or something, you have to explain them in very simple terms. And so one of my famous quotes is Albert Einstein says, if you can't explain it simply, you don't understand it well enough. And so that's something that you need to strive to do, because there's a lot of uncertainties there as well. The next slide will give you just one idea, one framework. There we go. This is out of the WHO, and this is just one type of uncertainty analysis. It's very general. And in this particular case, it's also qualitative. So you can, before you go into an uncertainty analysis, you need to take your data set and, even more importantly, define what is your problem. What are you trying to solve? You're trying to go with the skin formulation, and the problem that you're trying to you're trying to address is it good for sunscreen or something along those lines? It's something that you guys are more adept at than I am as a risk person. So if that's your problem, then your uncertainty analysis is a little bit different, because the sunscreen is going to be protective. So there's now a risk trade-off. And so your uncertainty analysis might be different. This is just one general way to do it. And again, many different people have looked at different uncertainty analyses and have frameworks for it. Let's go to the next slide, Barry. We'll talk a little bit about data analysis. So back in 2009, we had in our country the US NRC, the National Research Council, a National Academy of Sciences, put together this framework. And the framework was pretty telling for most of us, because it was saying, we really need to get away from whole animal testing. Well, why? It's worked for us for a number of years. Well, because of the animal protection ideas that many people have now, including scientists that work with the animals. But more importantly, we have this whole wealth of knowledge when it comes to genomics and proteomics and gene arrays. And can't we look at the cell and have the cell tell us what's going on just by the way it upregulates DNA or downregulates DNA or does something else? And so this framework was put together, and we tried to fit within the framework our standard ideas of hazard identification or characterization, exposure assessment, mode of action, and all of these things we were able to fit them into an understanding of the general way we did these things. And the next slide will show you sort of the picture you've seen. Well, you haven't quite seen this one. I thought maybe we must have skipped one. But the idea of systems biology, you know, what are we going to use it for? Well, we're going to use it for all of these different things. And let's go to the next slide, because I think that shows the picture. Okay, this is another, well, we must have just missed it. The picture I was thinking that we had, and I apologize for not having it in there, is the NRC put together a very nice template of the adverse outcome pathway and the various things that you have to do to cause an effect to occur or have a reparative effect occur. So you get this impact on the cell, and the cell, yeah, it was the slide before this, Barry. I took it out. I apologize. So you get this impact on the cell, but the cell has repair mechanisms. So you get this iterative process and upregulation of protective enzymes. And so what you get is this iterative thing. And then the NRC put this slide together, and this slide just teases out what I've just said before. So let's just keep going on, Barry. I should have known that I left that slide out. So we're going to use this in different places. We're going to use this in hazard identification or characterization. And in certain areas, we have mature thinking here. So in generating a hypothesis, if we have a cell that upregulates something and we know what that something is, we can develop a hypothesis of what's going on with the chemical holistically or more in the whole animal. With the hypothesis testing, it's developing, and with this idea of endpoint identification, some of the endpoints we can identify pretty well, and some of the endpoints we don't know what's going on here. So if you look at risk assessment generally, body weight decreases are very sentinel for whole animal studies. Well, how do you do endpoint identification of body weight decrease in silico? Well, we're not sure how that works. And in a dose response assessment, in like fashion, we've got some developing ideas, and we have some ideas that are still perhaps a little bit immature. And an exposure assessment, I would say most of this stuff is immature, but maybe not. Maybe I'm a little bit behind the times. How about the next slide, Barry? And we'll go to that. We've already seen this. Biologically based dose response models, if we've got a full one, which is very rare, that's wonderful. Biologically motivated models are helpful, but what we're really going for here is this idea of using precursor information. This is the in silico data that we're getting, precursor information, and doing dose response information or dose characterization or risk assessment on the basis of the precursor data. And this is new to most risk people. I've published at least one paper, or our group published at least one paper with acrylamide, where we actually folded in some in silico data to our understanding of the mode of action. But we used whole animal data for the assessment. We were still not able to just use it in the genomic data for the assessment. Okay, next slide. I think Barry will give us a little bit of issues and challenges. Okay. What is needed for all this in silico data? And some of the data that you have for skin is phenotypic anchoring. What is the definition of an adverse effect? And now, perhaps for the skin organ, we have this better worked out than we do for other organs. Characterizing the cell pathway. Well, we have adverse outcome pathways that Barry showed earlier that we are starting to build up for these endpoints in the skin. So that looks like it's well on the way for skin. Additional markers more generally, yes, we do need additional markers more generally. And we need models for different kinds of ICIDs. So if you have endocrine effects, which are multiple to different endocrine organs, or if you have immunotoxicity, and that's different for skin as relationship to other organs within the body, how do we match these different kinds of in silico findings to the various toxicities that we want to prevent? Of course, the in silico doesn't always have lack of metabolism, and we've already had several of you talked about, or several of our speakers already talk about volatiles. How do you deal with volatiles? How do you deal with mixtures? The next slide, I think, will give us another set of challenges, at least the risk assessment challenges here. Exposure duration. Well, of course, what we're interested in with any kind of chemical application to the skin, if it's a sunscreen or something like that, if you live in Alaska, you're not going to get a lot of sunscreen put onto folks up north because they don't have the need to do that. But if you're somewhere down south, you might have to use this skin salve multiple times during the year, multiple times during the day. And so the impact of exposure duration is important. We really don't have good ways to go in vitro to in vivo extrapolation. Now, maybe with the ex vivo, the skin arrays that are using real skin, but in an ex vivo situation, that might be more easy to do the extrapolation. But with a lot of the organs, we don't have that. Population variability. Well, that's true of all of us. We are all wonderfully variable, and there's nothing wrong with the variation. But what risk assessors have tried to do traditionally is say we're going to protect the most sensitive individual or sensitive subpopulation. And you'll get risk assessment people throughout the world that will say we don't protect the absolute most sensitive individual. So there's idiopathic responses in individuals. We don't try to predict that or actually try to protect that unique sensitive individual. What we do is we try to protect sensitive subgroups. And so the population variability, if you can characterize it, and in your case, if you can characterize the individual variation in skin sensitivity amongst different humans or different types of humans, young versus older, that's really going to be important. And of course, in ecological as well, the human focus, well, we focus on humans, that's fine, but there's also ecosystems. And if we could do in silico and predict ecosystem health, that would be wonderful, but it's still a challenge. And so with that, I think I'll end. I think there isn't any other slides there, Barry. So I'll pass that back to you, and then maybe we can have a discussion overall about how to go forward in this interesting area. Thank you.